Glands are formed by the epithelial cells, and we are going to discuss several types of the glands. In one classification, you can mention the glands could be three types, like the serous type of gland, and the other is the mucinous type of gland, and then we also have a mixed variety which is seromucous. Then there is one more classification, and it will be talking about the merocrine variety, apocrine variety, and the holocrine variety. Actually, we'll be starting with this classification only. We want to discuss the merocrine variety, which is also called as acrine variety, and then it is the apocrine variety, and lastly we will be talking about the holocrine variety. So let us begin with this classification, and then we can move on to the next as well. Here in this diagram we are looking at the salivary gland, basically the parotid salivary gland, and it is chiefly merocrine variety, which is also called acrine variety. When we talk about the mammary gland, it is a modified apocrine type of gland, and as it comes to sebaceous gland, it is holocrine variety. Let us focus upon the very first, the parotid salivary gland and the merocrine variety. What are the characteristic features here? In the first type, the merocrine variety, we have to understand that the secretion is coming out of the cell, but the cell membrane is intact. There is no loss of cell membrane, and only the secretions are moving out. So this is the merocrine or acrine type of glands. One example we have taken, parotid salivary gland. Let us move on to the next one, the apocrine variety of glands. We are magnifying this one, and in the apocrine variety, as the name is telling, apo, the apical. The apical portion of the cell membrane is broken down and is covering the secretion to form some blebs. Like one example is lysosomes. Whatever secretion the cell is having is being covered by a portion of the membrane, apical portion of the membrane, has the name apocrine, and some blebs will be torn out from the cell. So there is definitely some loss of cell membrane. In the apocrine variety, as we have to compare it with the sebaceous gland, the holocrine variety, holo means whole. The whole of the cell membrane is getting compromised. As we see here, the holocrine, the wall of the cell membrane is broken. Even the cell organelles are broken and become the secretions. So this is what you find in the sebaceous gland, the holocrine variety, where you see all the constituents of the cell become the secretions. In this case, it is the sebum, which comes on the skin for lubrication of the dry surface. Now, these were the schematic diagrams. Let us look at the actual histological pictures. Here we find the three different types of glands. First one, the merocrine. The second one is the apocrine, and third is the holocrine variety. As you notice, in the holocrine variety, the cell membranes will be intact. Only the secretions will be coming out, and there is no loss of cell membrane. So, as we see in the merocrine variety, these are the cells with intact cell membrane, and the secretions will be poured into the lumen of the gland. Whereas, if you are comparing it with the apocrine variety, you notice these are the cells, and their cell membrane is broken to cover the secretions, forming some of the blebs. We'll be drawing this diagram ourselves as well, but this is the type we are calling it as the apocrine variety. As you can notice, these are the blebs having some of the secretions given by the cell, covered by the cell membrane. Apical cell membrane is broken to cover the secretions. And then we look at the third one, the holocrine variety. The example we have taken for the holocrine variety here is the sebaceous gland. You can see one hair follicle here, and along with the hair follicle, this is the structure of the sebaceous gland, where you will notice the cells in the periphery of the glands are having intact cell membrane, but as we come towards the duct of the gland, the cell membranes are broken, the organelles are broken, and everything is turning into sebum, the secretion. So here in the holocrine variety, whole of the cell membrane, the organelles are broken to become the secretion, the sebum, and which is then being poured in a duct along with the hair follicle, and this is what we call as holocrine gland. Let us draw these diagrams ourselves as well now. As we notice in the merocrine variety, this is the gland, and we are showing the cells here with intact cell membrane. Only the secretions are coming into the lumen of the gland with no loss of cell membrane. So that is the merocrine variety with intact cell membrane, only secretions in the lumen. Whereas when we are talking about the apocrine, the apical portion of of the cell membrane will be broken. We are showing one of the cell here and another one here, along with the secretions at the apical portion. And then we understand the apical portion of the cell membrane is broken to come out in the lumen of the gland as blebs. We have mentioned, let us draw them here. A part of cell membrane, apical portion along with the secretion, and one example is the lysosome, and this is the apocrine variety. Then let us draw the holocrine variety as well, where first we can show a hair follicle, and along with hair follicle we are having the duct of this gland, which is the sebaceous gland, one holocrine variety of gland, where you find the cells at the periphery are having the intact cell membranes. But as we come close towards the duct, there is loss of cell membrane. Even the organelles are lost. They are becoming the secretion sebum, which will be going along with the hair follicle. So the point here is, examiner will be giving you slides and putting a pointer here. You have to train your eyes to identify the sebaceous gland or the apocrine type of gland, plus this is the merocrine variety. Let us look at one MCQ now, asking us which of the following is holocrine variety of gland. And as we have learnt, holocrine means whole of the cell membrane is broken, the organelles are broken, becoming the secretion of the gland, and we already know this type of a gland is found in the skin, and it is the sebaceous gland. Now, if you have found your answer, choice number D as the sebaceous gland, holocrine variety, what about the sweat gland? The sweat glands, choice number A, are two type. Mostly it will be acrine variety, but sometime there are some apocrine variety also, though the apocrine variety of sweat glands will be having rare occurrence. Now, if you say the apocrine variety of sweat glands are rare, can you give some examples? They are found in the axilla. They are also present in the perigenital area. And one more example you can talk about is wax gland in the ear, ceruminous gland. So as you talk about the various examples under the apocrine variety of sweat gland, they are found in the axilla, in the perigenital area. And one other example is ceruminous gland, modified apocrine sweat gland. And let me tell you one more modified apocrine sweat gland. It is the mammary gland. So in the breast you have the glands, and these are modified apocrine sweat glands. Then what about the pancreas? There we will notice that the pancreas is a mixed type of gland, endocrine plus exocrine, and the exocrine portion of the pancreas is acrine variety. And you must remember, what you call as acrine is the other name for the merocrine. Merocrine or acrine means the same, but as such in this question, if you are talking about the holocrine variety, we should keep our answer as choice number D, the sebaceous gland, secretion being the sebum. Then we look at the second classification of the glands, which is mentioning it as serous, mucinous, or mixed. Mixed means it is a combination of seromucinous gland. Now, if you're talking about the serous glands, then the secretions will be thin and watery consistency as compared with the mucinous, where it will be thick and viscous. So, the consistency of mucinous secretion is it is thick and viscous. Here we are talking about the consistency of the secretion, and then we understand it could be mixed variety also. Some examples can be taken. The parotid salivary gland is chiefly serous type, whereas if you are talking about the mucinous, the sublingual salivary gland under the tongue is chiefly mucinous variety. Then what about the mixed salivary gland? The mixed salivary gland is under the mandible, submandibular salivary gland, and it is the one which will be showing large number of serous demilunes. We have got a question for that. Let us look at the question here. Where do we get the serous demilunes? Now, before we talk about the structure of serous demilune, where are they present? And as you can see, we have already mentioned serous demilunes will be found in mixed type of salivary gland, and that is why we have to take our answer as choice number D, under the mandible. If you say submandibular salivary gland is your answer because it is mixed type of gland or seromucinous, what is the structure of this serous demilune? There will be actually a mucinous gland, and on that there is a cap of serous gland. So seromucinous will be more evident in a diagram. But be careful, not only the submandibular salivary gland is seromucinous, even the sublingual salivary gland is seromucinous, but it is chiefly mucinous variety. That means this question can have double answer, but B is a better answer than C. Are you trying to mention that the serous demilunes are found in the sublingual salivary gland also? Yes, it is also a mixed type of gland. Then why you are taking B as a better answer than C? Because the question is asking large number, and we have to understand sublingual salivary gland is largely mucinous mucinous variety. And what about the parotid salivary gland? The parotid salivary gland is chiefly serous variety. So despite the fact that serous demilunes can be found in two of the options, we'll keep B as a better answer than C because sublingual is having more of mucinous variety. Let us look at the histological pictures of these glands and it will become more evident. And what you find here is the serous type of salivary gland, parotid, is being shown here, whereas the sublingual salivary gland, which is chiefly mucinous, is being shown here. If we are talking about the submandibular salivary gland, it is mixed type of gland, pseudomucinous. It is the diagram which we find here. So let us first focus upon parotid salivary gland, which is the serous type of salivary gland. Chiefly, it will be having the serous cells identified by the fact that they are cuboidal cells and they are darkly stained, as you are comparing them with the sublingual salivary gland, which is chiefly mucinous. So as you see, the sublingual salivary gland is being shown here, parotid is here. We are trying to compare the parotid salivary gland, which is serous type of gland, with the mucinous type of gland, the sublingual salivary gland, the gland under the tongue. And if you compare here, these are columnar cells and lightly stained cells. We have mentioned serous are cuboidal, whereas the mucinous are columnar. The serous cells are darkly stained, whereas the mucinous are lightly stained. It is because of the mucus contained inside them. So they are columnar and lightly stained? Yes, because of the mucus. And what about this submandibular salivary gland? It is mixed type of gland and you will find it is having the features of cuboidal, darkly stained serous cells. Along with that, scattered glands are of mucinous type, the columnar with lightly stained features. Let us draw these diagrams along with the histological picture ourselves as well now. Now the first variety which we are drawing is the parotid salivary gland, where we can focus one of the serous follicle here, and as you magnify it, it will be some pyramidal shaped cell or cuboidal cells, as identified by the fact they'll be having a central spherical nucleus, a feature of cuboidal cells. So the serous acinus, as you see here, will have cuboidal or pyramidal shaped cells, central spherical nucleus, and the secretion will be poured inside here in the lumen, and these cells are darkly stained if you compare them with the mucinous cells acinus. Now let us draw one mucinous acinus as well. Example, we have taken sublingual salivary gland, chiefly mucinous, and you will find that here the cells are tall columnar cells with oval basal nucleus. That is a typical feature of mucinous acinus, and these are lightly stained. So the mucinous acinus is being drawn here, columnar cells with the oval basal nucleus, and the secretion which you find here is basically mucinous, and that is why these cells are lightly stained cells. The structure will become more clear when we are looking at a combination of pseudomucinous cell. Let us magnify this diagram so that we can talk about serous demilune as well, the submandibular salivary gland, in higher magnification. And here you notice this is a mucinous acinus on which we find a serous demilune. So this is the serous demilune, and here is the mucinous acinus. Cells which are tall columnar cells with the oval basal nucleus, as you find tall columnar cells with oval basal nucleus. And these cells which are columnar with oval basal nucleus is making the mucinous acinus. On the top of that disc is a cap of serous gland. And how do you identify the serous cells? As you can see, they are cuboidal cells with central spherical nucleus and just sitting on the mucinous acinus like a cap. This is what we call as serous demilune. So here is the central spherical nucleus, as you will find here in the cuboidal cells. I have mentioned to identify a cell as cuboidal, you look for the central spherical nucleus, and to identify one columnar cell, look at the oval basal nucleus. So these columnar cells giving the mucinous secretion, lightly stained cells. Over that there is darkly stained serous acinus, the serous demilune. And this kind of a feature is mostly found in the submandibular salivary gland, though it can also be seen in the sublingual salivary gland, but in small numbers, since the sublingual salivary gland is chiefly mucinous. And what about the parotid salivary gland? It is chiefly serous. The consistency of the secretion is thin and watery, whereas the consistency which is the secretion from sublingual salivary gland will be thick and viscous. Having few serous demilunes, mostly serous demilunes are found in the submandibular salivary gland.